so you said let it be a good day for you people. So try to be engaged, try to be interactive. So welcoming Miss Ishwari to hand over the class so that we can start. Okay, we
Yes,
can start.
sure.
Okay, thank you. We can start the class.
I'm audible, right?
Yes, you are audible. You are audible. Audible.
Yes, sure. Okay. Yes. Good evening, everyone. As per as yesterday's follow-up session today, and I will be taking over the workshops right from the beginning, that basis, because I would want everyone to follow the page. That's it. Right. I hope the screen is visible.
Yes, yes.
Yes.
Possible.
Okay. Yes. So to begin with, next generation sequencing analysis, what is exactly sequencing is, this is one of the sequencing technologies that has come up. It's nothing new, but it's just that now it's much more widely used in a much wider scope. So next generation sequencing technology basically is, it will take the biological sample, it will extract the material. It can be anything, DNA or RNA, and the samples can be any samples like blood sample, saliva sample, a tissue sample, or bone marrow sample. And from this sample extraction, they further go for library preparation. Now for any library preparation here, in the sense, for any next generation sequencing to work, we require a head start. Sample alone cannot be used in next generation sequencing, so we need to amplify the sample. To make it more, we need to add some adapters, and we need to add DNA fragments, the silico materials. So that step involves library preparation, and in library preparation onwards, we get to amplify and add the adapters. After that, we also can check for the quantity and quality of DNA or RNA, whatever is the material. And then we can take it forward for sequencing. Once the second step first step in the second step, we are sure that we have enough quantity and good quality of DNA or RNA in the sample, we proceed with the sequencing. So in the sequencing machine, what it happens is, it takes the sample and it determines the read bases which are present in the sample, and it gives us the output. And the last one is the analysis part, which the bioinformaticians play take forward with. So analysis is where we get the data which the sequencing machine has generated, and then that data has been used, and then we convert it into fastq files. I will explain how we get the data from sequencing machines and how we obtain it in the fastq format, and then how we use it for further analysis. This is just the generic workflow in step 4 image, how fastq, which is the raw data, is used and in further pre-processing and further analysis step. This is the general workflow for all next generation sequencing technologies. So another point I should mention here is, so the bioinformatics or bioinformaticians do not have a role in any of the step 1, step 2, and step 3. We just have to, in terms of bioinformatics, we just have to keep in mind what is the sequencing machine used, what is the library prepared, so that we can also have an idea for what tools or what pipeline accordingly we can design our own workflow and proceed further. Because some are Illumina sequences, some can be Oxford, or some can be packed bio. So we need to know the knowledge about it. Other than that, we do not play a role in either preparation of a DNA sample or preparation of libraries or sequencing, putting the samples in sequencing. We usually are not much involved in that, but we just have to make sure that we know what library the sample we will be analyzing for, and what is the starting material, and what is the industry purpose for the analysis. Now, as I mentioned, there are different types of NGS sequencing platforms. Illumina sequencing is the most widely used one because it has been giving very optimum results. It is very cost effective, and that one is mostly accessible also. And Second one is iron torrent. So unlike the Illumina sequencing, this iron torrent, the difference is in identifying, in the technology, in identifying the nucleotide bases. Like Illumina sequencing uses fluorescence based technology to identify the nucleotides, but iron torrent uses a semiconductor technology. Then Beijing Genomics Institute and MGA Tech Company developed another sequencing machine which is similar to Illumina for NGS sequencing, that is a complete genomics technology. Then comes the current trend ones, that is the third generation sequencing, which is Oxford and PacBio. These are all long read sequencing technology. So Illumina basically doesn't give us more than 150 base pairs, the read length, but Oxford Nanopore and PacBio give us more than 250 base pairs, for example, and highest is 20 kilo base pairs read length, which these machines give. This is the currently newly developed one, which not many of them use, but nowadays they are using for whole genomic studies, like for example to sequence a tiger, which was never done before in India. So they have used pack biotechnology, sequencing technology. So to us, this is referred because long read lengths make sure you do not miss out any sequences in between. In Illumina, you have only short restricted reads, right? In long reads, you may not miss out some reads, and you can also identify new variants. That is another advantage of the new generation technology. So Illumina sequencing, it offers both paired and then single end reads. So here I have put an example of paired end reads, whereas single end, as the word itself says single, as we know, read goes from 3 primer to 5 primer. The is single end sequencing, it will only go from 3 to 5 primer end, but paired end it will also go to the 5 prime to 3 prime as well. So there is an advantage in using paired end files is because we will not miss out most of the data. In case like we have a single end read, there is a high chance we might have missed out some data while this tech machine was amplifying it. But pair end sequencing will make sure that we do not miss out the data. So pair end is always preferred over single end data sequencing. Any doubts until now?
I have one if I'm allowed to ask.
Yes, yes, please.
Uh, a very basic question from the first slide. Uh,
Yes.
I mean, why do we need to construct a library?
One second. Yeah, step two, right? Yeah.
Yes.
So, any sample you take, okay, be it a saliva sample or be it a blood sample,
Hmm.
it need not be 100% sure that entire sample has good enough data, DNA data or RNA data. While you are extracting it, the extracting the extraction process might even kill the DNA, or might contaminate your sample also.
Hmm.
And for machine to process your data, it requires very high input data. To get this high input data, we cannot like give lot of samples, right? So what we do, we take the samples, we amplify it, that is we basically like clone the samples using artificial primers and ligands. So they get attached to the sample, and it first, suppose there is one input sample, it will make the sample 50 times or 100 times of it, like 200 times of the sample, and then it will add its own adapter sequences and it will process. So here sequencing machine will have lot of high input data, so we won't be losing much of the sample here, in case because this is not a cheap process. It requires lot of effort, and money also lot goes over here, so many people make sure the library preparation is done in such a way.
Okay. I mean, can I express what I'm understanding?
Yes, yes, please.
Okay, so I mean, does it mean that because the DNA molecule which is to
Hmm.
be fed into this entire process
Yes,
is quite precious, so
yes,
we need to make many, 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 many copies of that
yes,
molecule,
yes.
and that this multiple copying of the DNA molecule is what is known as library preparation.
Yes, yes. It is one of the step in library preparation which the machine itself does, where you add the adapters, you feed in the adapters and ligations,
Hmm.
and it will only carry on forward with its amplification. The same, the DNA replication. which we see in the cell, the same happens in this as well. It just replicates the data and then it just ligates the adapter sequences.
Okay, because the word library, no, that was imparting a meaning like much more of something is going to have to be built up over there. While it, I mean, but it is not so. I mean, you made it clear that only that particular DNA molecule, which is to be fed
Yes,
into the
yes,
process,
yes,
that
yes.
only that will be replicated many times.
Yes, yes. And there will also be RT-PCR. I'm pretty sure you are aware of the word RT-PCR. So
Hmm.
the same principle happens here. There'll be one strand, and the in the amplification process, what happens is the primers which we add, they will make sure that it replicates the strand in the reverse strand as well. It copies the information in the
Hmm.
reverse strand as well.
Hmm.
So the original data can either be found in the single strand, the first original strand, or in the reverse strand of it.
Okay.
Okay, so this is like a library preparation principles. I mean, you can go through amplification and ligation in library preparation. So, and each is different. Like this is a general NGS sequencing technology I was explaining.
Thank you, ma'am.
In,
You made it quite clear, quite nicely. You made it clear, ma'am. Please clarify something more about that paired and the other one. That
yes,
was,
sure, sure. I'll just repeat it again.
yeah.
So, as I was saying, I'm pretty sure you're aware of the directions on the DNA strand. It's always from 3 prime to 5 prime,
Hmm.
right?
Yeah.
So our DNA strands, they are in 3 prime to 5 prime, and the reverse strands are made in 5 prime to 3 prime, right?
Hmm.
Now, taking the same example, sequencing also follows this principle. So single end will only let the strand be read only in one direction, that is from 3 prime to 5 prime. That's
Okay.
it.
Okay.
And paired and sequencing technology, what it will be doing is it will not only read in single end, that is 3 prime to 5 prime, but it will also read in the reverse direction again. So, like we have one strand, we are getting the same data twice, but in the reverse complement form as well.
Okay.
So we are, in case sometimes what happens is while the machine is reading the strand from 3 prime to 5 prime, it might miss out some data
Hmm.
which has attached on its reverse strand. So to
Hmm.
get that data, the paired and sequencing enables to get it,
Okay.
because you're sequencing the same strand twice.
Yeah. Yeah.
Yeah. So we have information over there, and we're not losing data over there.
Okay.
Yeah, that's the principle of single end and paired end.
So to be on the safer side, I mean, you said that paired end is a better thing than the
Yes,
single one.
yes, yes, yes.
Okay. Thank you, ma'am. Thank you.
Yes, yes, welcome. Even though, like in cost-effective purposes, also even though single end is very cheap, the people always prefer for paired and sequencing. You know, in the industry level, in the research level, they always go for paired and sequencing analysis
Yeah.
because they don't want their efforts to go for waste and everything. Paired
Yeah.
end is always preferred over single end.
Thank you. Thank you. Yeah.
Now to get started with the data, now we know how we obtain the data from the machine. So, so from step 3, we directly do not get fastq files. The sequencing machine has a particular file which is called as base call files. So the sequencing machine will generate the data in the file format called bcl, base call file. So it's basically a computer format file which will have all culminated data in it. These base call files are taken and they're converted into FASTQ files using the tool BCL to FASTQ. The third point. So in this step, what we are basically doing is in the library preparation, while we are loading the samples, they also add certain identifiers and they have indexed for each of them. So in demultiplexing process, which this BCL to FASTQ file does, it will separate each sample according to its identification numbers, and it will organize these files and it will generate all these files in the form along with the statistics like the quality, read length, and everything. And it will also give us a report for each of them. So before we process for further analysis and everything, the quality person will make sure all these FASTQ files. We directly do not get FASTQ files from the system, that is sequencing machine. we get it in base call file format. So most of them are not aware of this, so that's the reason I thought of mentioning here. Base call files, usually the quality analysis person handles this, that is conversion of base call files to fastq files using the tool base bcl to fastq. And for more information, I'll always encourage you to please go through Illumina sequencing page, which describes all of these steps in detail. Then going forward for analysis, there are certain files and file formats which are standard and general in NGS data analysis. As I was mentioning, the data, they are usually in FASTA or FASTQ files. So FASTA or FASTQ files contain each sample wise, each sequencing data along with the header information and the quality for each sequencing read. So either the raw data can be in FASTA file or it can be in FASTQ file. So these are the certain file recognitions that it can be either fasta.gz, that is a compressed FASTA file, or it can be just FASTA file which is not compressed, or FASTQ files and compressed FASTQ files. Then there are alignment files. Alignment files are generated when we use alignment tools like Botai tool or BWA tool, which I'll be talking in the next slides. These SAM and BAM files are uh, uh, alignment manipulation file. So SAM file is the one which we can use, and which it will contain all the information along with the coordinates of the map reference genome and sample genome. And BAM file is the computer readable file, so we cannot usually access BAM files, but it's just used for conversion and easy feeding to the analysis pipeline. And then come the variant calling files, that is the VCF file or BCF file. So variant calling files are only obtained while you do the whole genome variant calling analysis, that is whole genome sequencing analysis or whole exome sequencing analysis. Then in analysis like for RNA sequencing, we will do gene expression in that analysis pipeline. So those gene expression files are in text files, they are mostly tab delimited files. Uh, and some tools like cut adapt and star, they cufflinks, they have a different set of files which need not be referred to right now, because most of them have stopped using that analysis file, so I'm not going deep into that pipeline. Now coming to the basic raw data files, that is FASTA and FASTQ. Yesterday I remember mentioning we usually prefer for FASTQ files. Now there is a very basic difference and important difference between these two. Is FASTA file and FASTQ files, both of them, even though they have sequencing reads, FASTA file will not have quality scores, and FASTQ files has its corresponding quality scores. Example is this: the first file I have pasted is a FASTA file. So most important thing to remember is FASTA file starts with a header line that is a greater than symbol. It's a sequencing ID identification. So the system identifies that it is a FASTA file because of the sequence greater than symbol. And the first line is the header line. That is, it has all the information, that is sample information, lane information, and organism name. So, and this, after that onwards is a normal sequence. This is the sample sequence, nucleotide sequence it contains. And the FASTQ file here is, it also starts with an identification, but along with that, after that, it has, even though it has sequence line, next line will be an sign and an identifier, which we need not worry about. Then the last line you can see is quality scores. So the second line is the sequence, nucleotide sequence, and the fourth line contains the quality score corresponding to the above sequence. So, like over here, we know that quality scores are nothing but Fred quality scores. Now, Fred quality scores are nothing but the probability that the nucleotide sequence called by the machine is correct. Like, how much is the probability that that is a Fred quality score which is given. So here we need to get to know the codes. Here we need not by heart them, but usually we need to have an idea about, in case like here for example, the red identifier here, T has a Fred quality score which is greater than which is a bracket, so that is 29, which is basically a very low quality score. So FASTQ file has these quality scores, which is very important for all the analysis we do in NGS, because most of the tools do not function with FASTA files. They require FASTQ files because they take into consideration the quality scores, and then only they process and give us the result. Else most of them end up throwing up errors, and will not be compatible to perform further. Of course there are tools which will help us to convert faster files to fastq files, but those are false quality scores. We should not rely on them. And in case you're looking forward to publications and stuff, they make things little difficult. Then Linux, coming to Linux, most of the NGS data analysis happen properly on Linux because the pipelines tools, they're all coded using Perl language and Linux system, which is much accessible, and it helps us to perform multiprocesses in a simple way without any hampering. Yes.
Miss Ishri, can you just check the chat box? A lot of questions are there.
Yes. 1 second. Uh, Miss Preeti. Yeah, so Miss Preeti, to answer your question, it depends on what type of sequencing you want for your study. Most of them go for Illumina sequencing, but if you want like long reads to study, you can also go for PacBio sequencing, or Oxford Nanopore sequencing. If you want like RNA sequencing expression, you can also go for Illumina sequencing. Not much of a difference, but it all comes down to how much you are willing to spend for the sequencing cost as well. So that gets decided from your end only. But most common ones is Illumina. Uh, does adapter have to be specific for different organism like the case in Sanger? No, adapter sequences are fixed based on the sequencing machine you're using. For Illumina sequencing, there are certain fixed adapter sequencers. Oxford Nanopore has its own adapter sequencers, and even primers are fixed by them. What kind of files generate after run from machine? Yes, after machine runs, as I was saying, they are BCL files, basic call files. So those are the files that get generated after Illumina sequencing machine has processed the data. Those base call files are converted to FASTQ files. We need to convert them to FASTQ files. Otherwise, for analysis, no tool as till today takes into BCL files. And let me also tell you, BCL files is a club of all sort of samples, and I don't think we need that. We need each sample and its own sequencing file in a separate format, so we have to separate them. That is the process called demultiplexing, which I was talking about. Miss Arya, I will be showing the FASTQ format process a little while later. Mr. Yogesh, the cutoff of red quality score is in Illumina is usually above 34. We do not usually prefer lower than that. 30 to 34 is a range set, but it is always better to have above 34. I would also request to please switch on the mic and ask in case. Yes. It will, Mr. Rohit. It contains all the run of the flow cell. So it will also have the information about adapters as well, because we add adapters when in library preparation. So demultiplexing is a very important step before we analyze, because there we get to delete the adapter files also. Otherwise, they will also be included in the analysis, which we don't want them. Any more doubts, or shall I proceed further? Hello. Shall I proceed, or please let me know? Yes. Yeah. Yes. So as I was talking about the Linux system, We will so Linux is a separate operating system, and Windows versions of 10 and above, they have a feature called Windows subsystem feature. You can use it, enable it in your Windows system, and you can install Ubuntu, which is a distribution of Linux. And you can set up your system, and you just have to enter the password and ID and proceed forward with the analysis. Or there is a process called dual booting your Windows system, which requires you to have a bootable pen drive or a disk, and you have to free up your space, and then you have to boot your Linux distribution in that. I did not introduce this to users before, is because it takes a lot of memory, one, and you have to make sure that your Windows system has a lot of backup. High chance you might end up losing your entire Windows data. So which was why, to make things easier, I had asked you all to use the Windows subsystem for Linux as well. Then Mac system, though it comes in Unix terminal already, it's much easier to use for Linux based data analysis. And then other than that, we of course need the raw data in FASTQ files. So if maybe you come from a research lab, you will have your own sampled raw data in FASTQ file format. Or if you are just practicing or learning, you can always download publicly available datasets from SRA database. So NCBI is one popularly used database. I will ask you all to please go to NCBI once, so that you get to know all the tools available. Yeah, here if you can see, you have all databases option. I hope everyone is following me. Please let me know if I need to pause. Hello. Hello. Is everyone following me?
Yes, ma'am, I'm following. Please go ahead.
Yes, yes. So in NCBI, as you can see, it has a collection of multiple databases. The basic one we usually learn in our college level is the BLAST one, that is to search similar sequences. So BLAST is one very basic tool. Uh, just a second. Here you can do protein blast, nucleotide blast, and etc. You just have to click and give in your sequences. And what blast usually does is it will basically align your sequence, and it will give out matched sequences and what organisms it belongs to. I'm not taking for in detail because I don't want to deviate from the main topic. And there are many other databases like Assembly, Bioproject, Biosample. Cleanvar is an important database for clinical samples and information of clinical samples. Domain conserved domains, database for gap proteins. Then DBVAR, this is a gene. So if you want to find out any genes and gene symbol and gene sequences, you can always look in gene. Then there is the genome. In this workshop, I will be taking you to genome database also, wherein you can download genome, completely sequenced genome in NCBI for, like, for example, human. GEO datasets is where you will find raw data. So, and you have SRA, which is within GEO dataset. I can also take you down. Then you have many protein-based databases like OMIM, clustered OMIM, and then protein-based databases. PubMed is like a library where all the published journals are available. You can just go to PubMed and search for any studies you want, and you can get an access to it. Then taxonomy, toolkits, SRA, SNP, all these are the different databases available in NCBI alone. Now to download raw database, I will take you through GEO database, that is Gene Expression Omnibus. So once we go to GEO, I would like everyone to do the same. We can always search for keywords. Now for our workshop, we were looking for whole genome studies. So I will type WGS, which is the short form for whole genome sequencing, and it will give me a pop-up with a hyperlink. So it's saying that there are 1,691 studies for whole genome. which I definitely want to. And it has a list of studies which have performed whole genome sequencing studies. On the left hand side, you can always select the series, samples, platforms you want. Some if you click on platforms, it will give me all microarray platforms as well, along with Illumina, which I currently don't want. Then it will also give, we can also customize the type of studies like expression profiling, methylation, genome binding, genome variation studies. Any studies as I want. I can also click on just other, that will give me something like whole genome sequencing studies. Then author, in case you know which author you are looking for, you can always add the author here. And then if you want particular tissue specific studies also, you can always customize your search, and you can go forward. On the right side there are other filters as well. And now moving further, in this studies I am looking for whole genomic sequencing studies. And let me tell you, whole genome sequencing studies, the raw data is usually of a very high database. Sorry, it will have very high GBs, because of course the name itself says whole genome sequencing. And if we take mammals in consideration, they also have very huge data sets here. Genome sequence download from the list of the genome sequence download will be giving you the genome of that organism. You don't want genome. If when you're looking for raw data, you don't want genome here. Genome comes from for when you want to align your raw data with the reference organism. So I'll also be introducing you how to download the raw genome sequence as well. Uh, yeah. So moving forward, you can click on any data you want to download. And like for example, I'm just using a chicken study data over here. So here it will show me, you can always read about the type of study they have done, the summary here. They also mention what is the library preparation. As I was mentioning earlier, we need to know the data about library preparation also, so that we are sure that we are looking for the particular this is what we are also looking for. And then these are the number of samples they have used in the study. And you can download the raw data which is available in SRA. Either you can click through SRA link here and then stepwise download each data, or you can also go to bio project. It will give a list of data which is present in the study. So because I this is like a basic which I want to teach you all, I'll always uh, ask you to click on individual sample, go to the SRA run column which is in the last table, and then click on the run. Now of course here the data is of 19 GB and it will take a lot of time. Just for example, I will just give you one easy to download data. Just a moment. In the meanwhile, you can also look through the other studies which have conducted whole genome sequencing, and you can please proceed with any of your interests. You can also download my studies, but of course, my studies will have a lot of much more data size than just. I hope everyone is following here, and I want you all to please try downloading one of the data. In the meanwhile, I'll be providing one sample data you all can use. One second. You all can search for accession GSE GSE 205178. What you all can do is you all can open GEO database and you all can just directly enter the database ID. You all can search in the search bar itself GSE. So this will give me a whole genome sequencing study of extracircular DNA of cerebrospinal fluid of medulloblastima, and the organism they have used is Homo sapiens. But we need not worry because when I had downloaded, it was just 4 GB minimum for each sample. And the main important thing is for studies like whole genome sequencing, RNA sequencing, whole exome sequencing, comparative studies. When we want to perform comparative studies, we will always want to have a normal sample and a tumor sample. So that helps us to understand what exactly is the analysis doing. Like in this case, we are downloading a normal sample, and we can download one patient sample or two patient samples. So when we perform the analysis and when we call the variance, we can always identify what the tool has, the results, and we can always compare it with the normal sample, and we can always give our conclusions about the variance. Yes, SRA, so SRA database, it helps us to download the raw data sets which each study has been uploaded. So whenever you publish your project in NCBI, it always asks us to upload our data sets as well. And this is a publicly accessible database. So when you publish your study, you also upload your raw data. Analyzed data is an option. These people have also uploaded, the study people have also uploaded raw data which is available in SRA. So you can always click on each sample to get a data. You can always go to scroll down the page of the sample, go to SRA option, select the SRA ID. Then when you scroll down, so here they have given all the information about the study, about the sample, and about the library. You can also always read and understand. So they've mentioned what is the library they have used, and this is an Illumina sequencing machine, Illumina NovaSeq. Then they have used paired and sequencing here, and then they have given a construction protocol all about the library preparation and the kits they have used and how they have prepared. Then they have given us the link for their raw data. The raw data is of 7 GB over here. So I will click on the raw data link here, and it will always take me to a site where it has metadata information about reads and information about the raw data files. So now I want to download the data. Now it has of course uploaded in the cloud of NCBI. So there are two cloud links. I can choose either of them. I'll go to what I will do. The easiest way for in Linux to download it is I will go to the directory. Now, in Linux, the word directory means folder. So whenever I mention the word directory, please don't be confused. It just means folder. You can always type the command pwd, that is present working directory, and it will show in which folder exactly are you. So whenever I want to create a new directory, I will use the command mkdir, that is make a directory, and I will just say raw data, for example. One of the rule in Linux is you cannot leave spaces. Linux will never take a word with space. So if I want to create a folder named raw data, I cannot give space between raw and data. So I have to insert underscore here. Now when I click again ls, it will display the new folder, that is raw data. Now to go into the raw data folder, I have to use change directory cd and give the raw data folder name. Now, because there is nothing in this, it's displaying nothing. Now, to download the data in this particular directory, I will use the command wget. This is to download the raw data in the folder. And you can see there's this link. This is the cloud link. You can copy paste this link in your terminal and click enter. This it will start with downloading the raw data. I hope everyone is following until here. I would appreciate if you could follow the same.
I mean, do you have some? place where all these links are written so that I could just copy and use them?
Yes, yes. I will just mention here. Uh, Link is for downloading data, or do you want a command?
Both of them, ma'am.
Oh, sure. I'll just
Like
copy the.
my case is that I am having an Ubuntu system on which
Okay.
I opened up the terminal, but I don't know what to download and I mean where from.
Yes, yes. So you can always create a directory called with the help of make mdkir command.
Hmm. Yes, ma'am.
Manually also we can do it as we do in this Windows system. You can always go to File Manager or Files, and it will give you an option like this.
Hmm.
Or if you are in your Windows system also, you can always go to the file you want, and you can always create a new folder.
Hmm.
Okay, and that way it will make a new folder. So you in terminal you have to make sure you are in that folder. So to make
Yes,
sure you
Ishwari,
can always.
yes, Ishwari, can you look onto the chat box? Some
Yes, sir.
comments
Yes,
are
sir.
there.
One second. Uh, yes. I am providing the commands. One second. And Miss Harita, after selecting the SRR ID, what should we do? So after you select the SRR ID, it will directly take you to this page, which has all the metadata information and data access information. So you can go to data access tab in that page, and you will get links. So there are two ways to download this. One is via terminal, which I was explaining. That is, you copy paste this link and you go to the terminal, and you just paste the link using wget command. And the other way to download this is by just directly clicking the link, and it will just download it for you. It will be in your download section. So the only problem here is when you directly download it via link, it might take some time, whereas Linux terminal download usually processes very quickly. So if the commands to download is, first thing, to make a new directory, you have to use mkdir, everything in small letters, and then to. So mkdir, for example, you can give a folder name raw data. So this will make a folder named raw data. Now you can always check for the folder by typing the command ls. This will list all the folders in your current folder or directory. I hope you all can see the folder you have created. Um, Can we use downloading browser? In Ubuntu, you can download via browser also, but it might take a little longer. You can always try it, and not a problem. Then, once you create and the new folder, you have to go to the folder. So you have to use the command cd space the folder name. This will change your current directory. So suppose you were in desktop folder, and in desktop folder you have created a new folder called raw data using mkdir raw data command. What will happen is it will create the data. Now you have to go to the folder to let the data happen in the folder. Or you can also use the other way around, but I don't want to mention advanced there. So what you can always do is to go into raw data folder, you just have to use the command cd, that is change directory, and mention your folder name. Instead of raw data, you can also try with your own just data or rough data, anything, so that you can always learn your ways. And now when once you have changed into your new data, that becomes your current working directory. You can always check it by using the command pwd, which is present working directory. So this, when you type pwd, it will give you something like this. It gives the entire path for your folder. Like my raw data is within home and Ishwari and then raw data. So similar way, even your folder will be suppose desktop and everything or C drive or anything. It will always give you the entire path of the folder. Now to copy to download anything, be it not just the raw data. In case you want to download and file, or you want to download. Let's talk about data only in this term. You have to use the command wget, which is the easiest command to remember, and copy paste the link. So here, how will I do is wget space. I have a cloud link over here. I'll take the cloud link. I'll copy from SRA database and I will paste it in the uh, terminal after listening, and I'll press enter. So it will take all the fetch the data and it will download the data in the current directory I am in. And it will give us a running time here. So for me, it has downloaded, if you can see my screen, it has downloaded 1.3 GB data and the current internet speed and what is the estimated time, like half an hour for downloading this current SRA ID, or 7 seconds based on how fast your internet speed is. I want everyone to please follow this. Either download directly or from Linux terminal, whatever way you're comfortable with. Kindly let me know once you're done downloading. When you yeah, and in case you get any errors such as command not found, you just have to install the command. Like here, I can just copy your error in Google, and it will give me one solution. I just can follow the solution. You can just install using do install, or you can always install using sudo command. One easy way, when any nothing works in Linux, you can also always use a sudo command, that is super user. Um, Miss Nathan, I would ask you to please install the wget command using any of the pages available. GitHub also has a solution. So, and everyone able to download the data? I'll display the command line commands here. Also, I have shared Google Drive link which contains all the basic Linux commands you can learn this for basic operations in Linux, and I have also given a beginner's book for bioinformatics analysis also. So, is everyone following this? Can I get some response? Hello. Uh, I hope everyone is following this. Okay. I shall keep the commands open for you. Please feel free to proceed and let me know once you are done.